Welcome friends to a new session on genetics. In this session, we will discuss theory of linkage and crossing over. Linkage and crossing over. An organism contains many characters in its body. Each character is determined by genes. An organism may have numerous genes for its various characters. The number of genes of an organism thus exceeds the number of its chromosomes because most organisms contain limited number of chromosomes. Hence, a single chromosome carries a large number of genes. Thomas Hunt Morgan, who is a Nobel Prize winner in physiology medicine in the year 1933, for his discoveries elucidating the role of chromosome in heredity, while he was performing an experiment with the Drosophila in 1910, he noticed that coupling or repulsion was not complete. He proposed that two genes are found in coupling phase and or in a repulsion phase because these were present on the same chromosome, which is known as coupling, or on two different but homologous chromosomes known as repulsion. Such genes are then called linked, and the phenomena is called linkage. He further suggested that the strength of linkage will be determined by distance between two genes in question. The greater this distance, the linkage strength will be lower. The linkage will be broken due to the phenomenon of crossing over occurring during meiosis. Crossing over will be relatively more frequent if distance between two genes is less. The phenomenon of crossing over involves exchange of chromosome segments. Let us first see about linkage. The linkage is defined as the tendency of two or more genes remain linked together in the original combination of the same chromosome during the process of inheritance for number of generations. Let us see about different types of linkages. The first one is complete linkage. It is the phenomenon in which two or more genes or characters are inherited together for a number of generations. In this, the genes are closely associated and tend to inherit together. Complete linkage is due to the fact that there occurs no break in chromosomes. As a result of complete linkage, the young ones inherit only the parental characters. New characters do not appear among the young ones. So, complete linkage produces only parental combination. New combinations do not rise. This phenomenon is very rare and found only in male Drosophila. Let us see in detail about linkage in Drosophila. In Drosophila, grey color is represented by capital G is dominant over black color represented by small g, and long wing represented by capital L is dominant over vestigial wing represented by small l. The genes for these characters are linked together in the same chromosome. When a fly with a grey body and long wings with the composition capital G, capital L and capital L is crossed with another fly having black body and vestigial wings with GGLL, all the F1 individuals are having grey body and long wings. The genetic composition would be capital G, small g, capital L, small l. When F1 male hybrid is test crossed with the double recessive parent, the F2 contains only two types of individuals in equal numbers. it is in 1:1 ratio instead of the expected 1:1, 1:1 ratio. The resulting offsprings are grandparents. This is because the gene capital G is linked with capital L and the gene small g is linked with small l. The F1 male hybrid is back crossed with a recessive female parent. The F1 male hybrid produces only two types of gametes in which the linked genes capital G and capital L or small g and small l are inherited together. So, only two types of offsprings are produced in the F2 in equal numbers. Now, let us come into the case of incomplete linkage. In incomplete linkage, the linked genes on certain occasions separate. This leads to the formation of new combinations among the young ones. Incomplete linkage is due to the breakage of chromosomes during gametogenesis. Incomplete linkage is found in female Drosophila. This breakage of chromosomes leads to the separation of linked genes and new combinations appear. Because of these new genic combination, the offspring produced in the F2 generation are different from their parent in their phenotype as well as genotype. So, the incomplete linkage involves the accidental breakage of chromosomal segments or linked genes, resulting in new combinations of genes. In the above experiment, the F1 female hybrid produces 4 types of gametes. Among 4 types of gametes carry new combinations due to the separation of linked genes. The capital G gene is separated from capital L and joins with small l. In the same way, the capital L joins with small g. These combinations are different from the original combination. This type of inheritance is different from the independent assortment. If the genes are assorted independently, 4 types of offsprings are produced in F2 in the ratio 1:1:1:1. There are different theories explaining the linkage. Let us see one by one. The first one is differential multiplication theory proposed by William Bateson in the year 1930. He assumed that the linkage is due to the differential multiplication of cells containing different combinations of genes. After the segregation of genes during gamete formation, the set of gametes possessing parental combinations multiplies rapidly than the set having non-parental combination. This theory was disproved because after segregation of genes, there is only one division. Secondly, let us see about chromosome theory of linkage. This theory was proposed by T.H. Morgan and William Ernst Cassel. This theory states that 1. Genes located in the same chromosome are inherited together and are said to be linked. 2. The linked genes are arranged in a linear fashion on the chromosome. 3. The degree of linkage is determined by the distances between the genes. Closely located genes show strong linkage, while genes which are widely located show weak linkage. Now, let us look into the arrangement of linked genes on the chromosome. Alfred Henry Strand, who constructed the first genetic map of a chromosome in the year 1913, found that the genes are arranged in chromosome in a linear manner. Based upon the arrangement of the genes in the chromosome, the linkage can be classified into 2 types. 1. Cis arrangement. In cis arrangement, the 2 dominant genes capital B and capital L are located on one member of the chromosome pair. The 2 recessive genes small b and small l are located on the other. This type of arrangement with the 2 dominants on the same chromosome is called cis arrangement. The heterozygotes with such arrangement are known as cis heterozygotes. The second one is trans arrangement. In this type, the dominant gene of one pair and the recessive gene of other pair, capital B and small l, are located on one chromosome, and the recessive gene of the first pair and dominant gene of the second pair, small b and capital L, are located in the second chromosome. The heterozygotes with such arrangement are known as trans heterozygote. This you can see in the following figure. Now let us examine about linkage groups. All the genes which are linked with one another form in a linkage group. Since linked genes are present in the same chromosome, the number of linkage group in an animal or plant is equal to the haploid number of chromosomes present in its cell. This hypothesis was given by Walter Stanborough Sutton, who contributed Boveri-Sutton chromosome theory, and was further proved by experiments on Drosophila by T.H. Morgan. In Drosophila, there are 4 linkage groups corresponding to the 4 pairs of chromosomes. Man has 23 linkage groups corresponding to the 23 pairs of chromosomes. The biochemical approach is based on the frequencies of recombination between markers during crossover of homologous chromosomes. The greater the frequency of recombination between two genetic markers, the further apart they are assumed to be. Conversely, the lower the frequency of recombination between the markers, the smaller the physical distance between them. Historically, the markers originally used were detectable phenotypes such as enzyme production, eye color, which was derived from coding DNA sequences. Eventually, confirmed or assumed non-coding DNA sequences such as microsatellites or those generating restriction fragment length polymorphisms, in short we call RFLPs, have been used. Now let us look into the phenomena of crossing over. The phenomena of complete linkage occurs rarely because the linked genes do not always stay together. Sometimes they are separated from one another during meiotic division, which leads to incomplete linkage. Linked genes separate at the time of break in the chromatids of homologous chromosomes, followed by mutual exchange of corresponding parts and interchange of alleles, resulting in the appearance of non-parental combinations. Morgan and his collaborators termed this phenomenon as crossing over. Crossing over may be defined as the interchange of chromosomal parts between non-sister chromatids of a homologous pair, resulting in recombination of genes. In detail, let us see about the mechanism of crossing over. Crossing over occurs during meiosis of gametogenesis. The homologous chromosomes move towards each other and come to lie side by side. This phenomenon of pairing of homologous chromosomes is called synapsis. The paired homologous chromosomes are called bivalents. The homologous chromosomes split longitudinally. Each chromosome split into 2 chromatids. Hence, 4 chromatids are produced from 2 homologous chromosomes. This stage is called tetrad stage. The 2 chromatids of a chromosome are attached by a single centromere. These are called sister chromatids. The non-sister chromatids of homologous chromosomes twist each other. At certain points, the non-sister chromatids are connected with each other. These points of contacts are called chiasmata. In singular, it is called chiasma. Chiasma means a cross. At the chiasma, the chromatids break. This is brought about by an enzyme called endonuclease. The broken segment of one chromatid is fused with the other chromatid and vice versa. This fusion is brought about by another enzyme called ligase. This exchange of chromosomal segments between non-sister chromatids is called crossing over. After crossing over, the non-sister chromatids repel each other. They separate from each other from twisting. The separation starts from the centromere towards the ends like a zip. This separation is called terminalization. This leads to the separation of homologous chromosomes. There are different theories put forward regarding crossing over. Let us examine one by one. The first one is chiasma type theory. This theory was proposed by Franz Alphonse Janssen, who is the discoverer of the crossing over of genes during meiosis called chiasma type P. This theory suggests that prior to crossing over, the chromosomes of each bivalent get duplicated to form tetrad. Crossing over occurs only between the non-sister chromatids of a tetrad. In the diplotin stage, the non-sister chromatids overlap with another and form chiasma or point of contact. In the chiasma, the chromatids break and they rejoin with the mutual exchange of segments. The other two chromatids remain intact. The second theory is the breakage first theory. The theory was proposed by Hermann Joseph Muller, who is a Nobel laureate for his work on the physiological and genetic effects of radiation. According to this theory, the non-sister chromatids first touch and cross each other. Then breakage occurs at the points of contact of the chromatids. The broken segments rejoin to form new chromatids. The third theory is the contact first theory. This theory was proposed by Alexander Sergeyevich Serebrovsky. According to this theory, the non-sister chromatids first touch and cross each other. Then breakage occurs at the points of contact of the chromatids. The broken segments rejoin to new combinations. The fourth theory is Strand or torsion theory. This theory was proposed by Cyril Dean Darlington, who was a fellow of Royal Society and who discovered the mechanics of chromosomal crossover. According to this theory, the homologous chromosomes are relationally coiled around each other in a specific fashion. The chromosomes duplicate and the two sister chromatids in each chromosome are also coiled one around the other. When the chromosomes start separating, their relational coiling unravels in one direction and the sister chromatids unravel in the opposite direction. The repulsion causes torsion, which exerts pressure or strain on the weak non-sister chromatids. The non-sister chromatids break at the point of contact and broken ends rejoin and form chiasma. The fifth theory is differential contraction theory. This was proposed by Charles Leonard Huskins. He suggested that tension set up by differential contraction after pairing may cause breaks at overlaps. Now let us see about kinds of crossing over. Based upon the number of chiasma formed, crossing over may be classified into three. The first one is single crossing over. In this type, only one chiasma is formed. Only one chromatid of each chromosome is involved in single crossing over. The second one is double crossing over. In this type, two chiasmata are formed between the same chromatids or different chromatids. Thus, two or three or all the four chromatids of the homologous chromosomes may be involved in the process of double crossing over. This is depicted in the following figure. The double crossing over is again divided into three subtypes. The first one is two-strand double crossing over. In this type, two non-sister chromatids are involved at both the crossing over points. Here, two chromatids are non-crossovers and maintain the parental combinations of genes. The second is three-strand double crossing over. In this type, three chromatids are involved in the formation of chiasmata. Here, three chromatids exchange their parts and one chromatid is non-crossover. and the third one is 4-strand double crossing over. In this type, all the 4 chromatids are involved in the formation of chiasmata. Now, let us look into the multiple crossing over. In this type, more than 2 chiasmata are formed. It is very rare because of interference in crossover. Other 2 types of crossing over are germinal or meiotic crossing over and somatic or mitotic crossing over. Normally, crossing over occurs only in the germinal cells of reproductive organs during the process of gametogenesis is called germinal crossing over. The somatic crossing over produces patches of crossover tissue between the normal cells and they are present as mosaic appearance. Now, let us examine proofs for crossing over, 2-point and 3-point test cross. In 1910, T.H. Morgan and his students extended the concept of the chromosome theory of inheritance. By carefully analyzing the results of crosses involving Drosophila, Morgan and his students demonstrated that genes are arranged in a linear order on each chromosome. They also demonstrated that independent assortment does not apply if the 2 loci lie close together in the same pair of homologous chromosomes. In Drosophila, there is a locus controlling wing shape, the dominant allele capital V for normal wings and the recessive allele for vestigial wings. Another locus control body color, the dominant allele capital B for grey body and the recessive allele small b for black body. If a homozygous capital B, capital B, capital V, capital V is crossed with BBVV all in small letters, the F1 will have grey bodies and normal wings represented by capital B, small b, capital V, small v. Because these loci lie close to each other in the same pair of homologous chromosomes, their alleles do not assort independently. Instead, they are linked genes that tend to be linked together. We can observe the linkage in the above test cross in which heterozygous F1 flies represented by capital B small b, capital V small v are mated with homozygous recessive flies B, B, V, V all small letters. This is clearly depicted in the following figure. The heterozygous individuals are mated with homozygous recessive individuals. This test cross is called a 2-point test cross because the alleles of 2 loci are involved. If the loci controlling these traits were unlinked, the heterozygous parent would produce 4 types of gametes: capital B, capital V, capital B small v, capital V and small b small v in a test cross in equal numbers. Due to independent assortment, it would produce new gene combinations not present in the parental generation. Thus, in F1, 25% of the offspring would be grey normal represented by capital B small b, capital V small v. 25% is grey vestigial represented by capital B small v, 25% is black normal wherein the B, both B is small letters, then capital V small v, and the rest 25% is black vestigial, all represented by small b and small v. By contrast, in our example, the alleles of the loci do not undergo independent assortment because they are linked. In our example, 217 of the offsprings are recombinant types. Grey vestigial with genetic constitution capital B small b, VV small letters is 111 numbers, and black normal with genetic constitution small b small v, capital V small v is 106 numbers. The remaining 1051 offsprings are parental types. We can calculate the percentage of crossing over using this data by adding the number of individuals in the two recombinant types of offsprings divided by the total number of offspring and multiplying by 100. Thus, B locus and V locus have 17% recombination between them. A 3-point test cross involving 3 genes give us information regarding relative distance between the linked genes and also tell us the linear order in which these genes should be present on the chromosome. So far, we have looked at linkage in crosses of double heterozygotes to double recessive parents. The next level of complexity is a cross of a triple heterozygote to triple recessive parents. This kind of cross, called a 3-point test cross, illustrates the standard approach used in linkage analysis. Here we focus on 3 Drosophila genes that have the non-wild type alleles small s, small c representing scute or loss of certain thoracic bristles, small e, small c representing echinus or roughened eye surface, and small v, small g for vestigial wing. We can cross SC, SC, EC, EC, VG, VG triple recessive flies with wild type flies to generate triple heterozygotes SC, SC plus, EC, EC plus, VG, VG plus. By test crossing heterozygous females with the triple recessive males, we can analyze recombination in these heterozygotes. 8 gametic types are possible and they were counted in a sample of 1008 progeny flies. So, we can analyze such crosses to calculate all possible recombinant frequencies. At first, we can see that there is a considerable deviation from the 1:1:1:1:1:1:1:1 ratio that is expected if the genes are all unlinked. So, let us begin to calculate recombinant frequency values, taking the loci a pair at a time. Starting with the SC and EC loci, we determine which of the gametic genotypes are recombinant for SC and EC. Because we know that the heterozygotes were established from SC, EC and SC plus EC plus gametes, we know that the recombinant products of meiosis must be SC, EC plus and SC plus EC. From the list, there are 12 plus 14 plus 14 plus 16 is equal to 56 of these types. Therefore, RF is equal to 56 divided by 1008 into 100 is equal to 5.5 mu. These frequencies tell us that these loci must be linked on the same chromosome as shown in the figure. Now, let us look at recombination between the SC and VG loci. The input parental genotypes were SC VG and SC plus VG plus. So, we must calculate the frequency of SC VG plus and SC plus VG progeny types. There are 243 plus 233 plus 14 plus EC 16 is equal to 506 recombinants, because 506 divided by 1008 is very close to an RF of 50%. We conclude that the SC and VG loci are not linked and are probably not on the same chromosome. We can summarize the linkage relationship as shown in the figure. Adding up the recombinants in the list and calculating the RF will confirm that the EC and VG loci must also be unlinked. Having made those deductions about linkage, we can rewrite the parents of the test cross as SC plus EC plus SC EC, VG plus VG into SC EC, SC EC, VG VG. Now, let us examine the statistical significance and relevance of 2-point and 3-point test cross. A cross involving 2 loci is usually referred to as 2-point cross. It gives us a powerful tool for dissecting the makeup of a chromosome. This analysis is to look at 3 loci simultaneously so that we can determine their relative order on the chromosome. More important, we can also analyze the effects of multiple crossovers which cannot be detected in a 2-point cross on map distances. 2 crossovers between 2 loci can cause the chromosome to look as if no crossovers took place, causing us to underestimate map distances. Thus, we need a third locus between the first 2 to detect multiple crossover events and more accurate values. Now, let us summarize the foregoing discussion of linkage and crossing over. Thomas Hunt Morgan, while performing an experiment in Drosophila, found that during the process of inheritance there is a tendency of 2 or more genes remains linked together in the original combination of the same chromosome for number of generations. Such genes are then called linked and the phenomena is called linkage. He further suggested that the strength of linkage will be determined by distance between the 2 genes. The greater the distance, the linkage strength will be lower. The linkage will be broken due to the phenomenon of crossing over occurring during meiosis. Crossing over will be relatively more frequent if distance between 2 genes is less. The phenomenon of crossing over involves exchange of chromosome segments and occurs during meiosis of gametogenesis. The linked genes are arranged in the chromosome in the linear manner. A test cross involving 2 loci called 2-point test cross and 3 loci called 3-point test cross gives us a powerful tool to analyze the makeup of a chromosome and crossover effects. Now, I am giving you certain assignments based on the discussions we had. The first one is describe 2-point test cross to detect linkage in fruit flies. Second is explain different types of linkage. Third, describe linkage groups. Fourth, write a short note on the mechanism of crossing over. Fifth, explain how the observations of genetics provide the basis for linkage and crossing over. I am also giving you certain references for your further reading. The first book, entitled Biology, 10th edition, is authored by Eldra, Charles, Diana and Linda and published in the year 2015. Again, another book entitled Biology, 8th edition, authored by Eldra, Diana and Linda, published in the year 2015. A third book entitled An Introduction to Genetic Analysis, 7th edition, published from New York, authored by Griffiths, Miller, Suzuki, Levantine and Gilbert and published in the year 2000. A fourth reference is entitled Chromosomes and Heredity, authored by Morgan Th. in the year 1910 and published in the journal The American Naturalist, volume number 44. The page numbers are 449 to 496. A fifth reference would be entitled Biology, 7th edition, authored by Campbell and published in the year 2012. I am also giving you certain website references for your further. The first one is www.genetics.org. The second one is theagricos.com. So, we have come to the end of a session. Thank you for watching this session and see you next time. Until then, bye.